Hey everyone, welcome to our monthly webinar. This month, we're going to talk about an integrative approach to genomics. We're going to talk about how to improve health, performance, and longevity by understanding your genomics. So we're going to cover several things today. First of all, we're going to go over the integrative approach to disease and disease versus health balance. We're going to talk about genetics versus genomics. We're going to have a little glimpse into the cell and how DNA is our blueprint. We're going to talk about how genetic variants impact us, and specifically single nucleotide polymorphisms or SNPs versus mutations. We're going to talk about how genetic variants and external factors play together. We're going to talk about the goals of genomics-based precision medicine. We're going to look at how we specifically look at your genomics to optimize health and performance, and a little glimpse into what a wild health clarity report looks like. We're going to briefly talk about how your mind can control your genes. And we're also going to show you how you can sign up to get your genomics tested. If you have any questions as I'm going through this webinar, please put them in the chat box and we'll get to them as soon as we can. All right, before we dive in here, I'd love for everyone to put in the chat box, what do you think our genes impact? And you can kind of keep doing that as I get started here, and then we'll kind of hear either Jessa or Em chime in with what people are saying. Anything coming in yet, you two?
Yeah, we have a few responses. They're still trickling in, but what we have so far is hair color, body type, cholesterol levels, how I look, lifespan, sleep, got heart disease, diabetes, got a couple people pointing out that they just affect our overall health. We've got brain function, likelihood of, a lot of people are naming specific diseases like hemophilia and sickle cell and cancer has come up a couple times. Height, intelligence, mental health, personality, fat metabolism, and mood.
Great, great. Those are all perfect. Excellent. All right, so we're going to jump into just looking at an overall integrative approach. So those of you who have watched any of the other webinars or been to them know that I always like to start talking about how integrative medicine is evidence-based, and it's basically the umbrella under which all forms of healing modalities with evidence exist. And so under the integrative medicine umbrella is genomics-based precision medicine, conventional medicine, regenerative medicine, energy healing, massage, sound healing, osteopathic medicine, et cetera. And it's important to note that all of these forms of healing modalities have evidence. So it's evidence-based medicine. If we zoom out and have a 35,000 foot view of disease versus health, it's really a balance. So we have our biologic systems, and then we have our internal and external stressors. Whenever our biologic systems are more robust than our internal and external stressors, we're going to be in a state of health. What are some of the biologic systems? Basically, all of our systems throughout our body, our adrenal, immune, detoxification systems, all those. Stressors are everything that's coming in all the time. So those include allergies, infections, things like toxins, stress, disrupted microbiome, all those things. We like to look at things holistically. So if I look at the entire body as a whole, then we can see that anytime we disrupt anything with biologic systems or stressors, it's going to impact everything in the body. From a conventional standpoint, which is where I worked for many, many years, we kind of divide it up into different areas. So if you have something going on with your throat, you're going to go to the ear, nose, and throat doctor. If you have something going on with your bones, you go to the orthopedic doctor. If you have something going on with your gut, you go to the gastroenterologist. And that works to an extent, but because everything is so interconnected, when we look at it that way, a lot of times we end up not helping to get the body in balance as quickly, if not at all. Now, overlying all these stressors in biologic systems is our genes and our genomics. So we're going to talk more about that, but I just want to mention that that's kind of an umbrella that impacts everything. And then if the internal and external stressors outweigh our biologic systems, then we kind of go into a state of disease and all the things that come with disease in terms of symptoms and diagnoses. And let's zoom in a little bit more. So again, for those of you who've watched this previously, watched other webinars, I used to talk about the 4 pillars of health, and I've recently expanded it to the 5 pillars of health. And the addition is social connection, which is so important. I wrote a blog article about this recently, about the impact of social connection on everything. And in some ways, there's probably more evidence for social connection than any of the other 4 pillars. So the 5 pillars: sleep, stress, nutrition, gut health, exercise, and social connection. We're always looking at boost, and I'm always looking at things through the lens of what is the root cause of what is going on. So, you know, 5 of us might all have diabetes, and depending on what's happening, maybe my diabetes is because I don't have social connection. Maybe yours is because you're not getting enough sleep. Maybe someone else's is because you're not getting enough exercise. So conventional medicine kind of treats everything pretty similarly in more of a Band-Aid approach. And we're always looking at what's causing it, and hopefully how can we get your body back in balance. And when we look at genomics with this, if we have genomic risks, and we're going to talk more specifically about what that means during this webinar, if you combine various different genomic risks with imbalances of those 5 pillars, that's where you get disease. And so things don't happen by themselves. We have to have a lot of different factors contributing for someone to end up in a state of what we would call disease. Okay, so the next thing we're going to move on to is talking about the difference between genetics and genomics. So genetics is a study of single genes and their role in the way that traits or conditions are inherited. And genomics is the study of an organism's genes, as well as its function and regulation. Genomics is what we're more interested in. Genomics kind of looks population-wide at how our genes are going to impact on a more broad level what happens within our body. Versus genes, this is pictures of myself with my mom and my dad. Genes impact how we look and how the traits are inherited, but they don't impact. The study of genetics specifically versus genomics is not as much about disease and how we combine what's happening within our genes with everything else in terms of the biochemistry and what ends up happening from the genes. And we'll talk more about that. But we're really looking at genomics-based precision medicine, not genes. We're not looking at how our traits look. The history of genomics is actually pretty recent, which is why probably a lot of you haven't heard much about it, haven't gotten your genomics tested. So the initial Human Genome Project, which mapped the genome of 20 people, was just in 2003. And then the 1,000 Genomes Project Consortium was just completed in 2015. So what that means is really it's only been the last 7 years that we can really use this in any sort of applicable way. And the other thing is we're constantly gathering more data with this. So this is a rapidly evolving field that is constantly improving, and there's not as much data as there would be if we were 20 years out from this. So sometimes people have concerns because of the lack of data, and that's completely founded, and that's just based on time, really. All right. So in order to kind of understand how genes impact us and how the study of genomics and looking at our genomics can help us with performance and health and longevity, we kind of have to go back to the cell. So this might be new for some of you. This might be scary for others, but I'm going to try and make it as simple as possible. So this is a little picture, a little drawing of a cell. And the purple part there is the nucleus. And the nucleus is kind of we'll say the brain of the cell in some ways, where our DNA is produced. And then the mitochondria, which you can see on the left, they look like little kidney beans. Those are kind of the little engines of the cell. And the mitochondria actually has its own DNA as well. And so when we're talking about how genomics impact us, we're talking about both what the DNA that comes out of the nucleus and the DNA that comes out of the mitochondria. DNA is made up of 4 different nucleotides that form base pairs. So there's adenine, thymine, guanine, and cytosine. And don't get overwhelmed by the sort of technical terminology with this. It's gonna, I just want to show this to everyone so you can kind of understand why variants make a difference. So you can see those little guys holding on to each other, the little base pairs there. And as they hold on to each other, that forms the double helix of DNA. And what's going to happen is, in order to for that DNA to actually make impacts on our body, our system is going to read that. So the DNA is almost like a blueprint. And DNA, DNA, believe it or not, in each cell, there's 6 feet of DNA, and there's 300 trillion feet of DNA in our body. So there's so much DNA that it actually has to be wrapped up super, super tightly in order for it to fit in our body. And that is important because the way that we talk about methylation, and the way that the DNA kind of gets packed up is actually with methylation. So methylation is really just a protein group that gets added to various processes throughout our body. And in this case, the methyl group is actually kind of keeping the DNA tight. And how that is important is that if the DNA is methylated and it's kind of held tight, then it's inactive. So the body is not going to read it to then make proteins. And then acetylation actually does the opposite. It kind of opens it up so it can be read. And this is just important to realize that, like, we have lots and lots and lots of DNA that is basically like a closed, locked book. So, if you have a big bookshelf of all your books and most of them have little locks on them, like kind of like old school diary locks, you're not going to be able to get in there. And all of the DNA, most of our DNA is like that. So our body's not actually using it actively. And if we have segments of DNA that potentially are going to code for things that are harmful, and they get opened up because they're not getting methylated adequately, that can actually cause problems and disease. So important to understand that, like, most of our DNA is not being used on a daily basis. And then the way it works is we have little sections of DNA, and that's what we call our genes. So if you can see in this picture here, those little sections of DNA then get basically turned into protein. And the process in which it works is through transcription and translation. Those are the terms, but basically our little engines and our little factories in our cells are reading the areas of the genes and then creating proteins from them. So that's really how DNA ends up impacting our body and our health. And then each of those little discrete regions that are called genes, they can range from pretty small 200 base pairs. And the base pairs there are, like in this drawing here, each of those are 4 base pairs. So they're going to range to be called a gene that's going to ultimately encode for a function. There will be between 200 to 2 million of those base pairs. And then humans have up to 25,000 genes. And then an allele, and again, we're just going over the terminology here, which will be helpful when we talk about how we actually look at this. So an allele is basically one of 2 or more alternative forms of genes that are found on the same chromosome. So if you look at here on the little drawings here, you may remember from high school or college or grad school biology that you can have either heterogeneous or heterozygous or homozygous alleles. So what that means is on the little diagram, the little drawing on the right here, if you potentially have 2 alleles for a particular trait, maybe you'll have it. If you don't have any, you're not going to have it. If you have one of each, you may or may not have it, just depending on other factors. And then you can see in the little diagram on the left with the chromosomes there that you're going to have 2 different alleles for each different particular gene, and it's just going to depend on if it's mutated or not, if you have variants or not, whether or not it's going to code or how it's going to code for something. So again, I don't want to get caught in the weeds too much here, but we will talk about when I go over the reports that you're going to get one of these alleles basically from mom and one of them from dad. And maybe you get, maybe you get 2 alleles that are going to make you predisposed to having vitamin D deficiency, or maybe you have none, or maybe you just have one, and that's going to impact your risk of having an outcome from your genes. And then just going back to how this works. So DNA, all those codes, all the different little sequences that I went over previously, DNA is going to turn into RNA, and it's all messaging basically. And then the RNA is going to basically allow us to create protein. So it's a blueprint. The DNA is a blueprint for the RNA, and then the RNA is basically a blueprint for the protein. So I think of it kind of like this diagram, like a little factory. So the DNA is hanging out in the nucleus or the mitochondria, and then our cells can read the DNA with messenger RNA, and then the messenger RNA basically allows the cellular factory to produce proteins. And that's what it all comes down to. So we're basically a big hunk of protein in many ways. And depending on how the proteins are working, we may or may not develop diseases. We may or may not feel strong. We may or may not get illnesses. There's lots of different types of protein structures. We're not going to get into the weeds here very much, but just suffice it to say that, when we're initially making the proteins, they're going to be a very simple primary structure, and then they're going to form together with other proteins to form more complicated structures that end up making our organs and everything about us. What do proteins do? They pretty much do everything in the body, but I think it's worth it to talk about some of them in the context of like, how are our genes impacting our health? So proteins are going to make our digestive enzymes that help facilitate absorption of nutrients and help us to digest our food. Proteins make antibodies and help with our immune function. Proteins are going to help with our DNA and RNA regulation and expression. Of course, we know proteins are going to help with our muscles, and we have muscles throughout our body everywhere. So, our external muscles that we think of, but then of course also our muscles inside our vascular walls and all that. So we need the protein for that. Protein also is going to just provide support for our body. If we didn't have protein, we wouldn't have any sort of support in our body. Protein are the building blocks for hormones. We know that hormones are signaling molecules that impact everything. And then of course, proteins are going to move essential molecules around the body. So if we don't have proteins, we're not going to be able to transport our oxygen and all the other important vital nutrients and molecules to health and longevity and performance and survival, frankly. So proteins are important for everything within our body. And we go back to that disease versus health model, the seesaw there. If we, in terms of our biologic systems and the robustness of our biologic systems, we need to have adequate, well-functioning proteins in order for our biologic systems to work well. And in order to have those optimal proteins, we need to have the DNA blueprints that are working optimally for us. So now let's talk about what happens if we have genetic variants, and how does that impact our proteins, and how does that impact our health? So important to recognize that 99% of all of us, of all of our DNA of all humans, are identical, not just identical twins here. And as I mentioned before, we have up to about 25,000 genes and 3 billion nucleotides. So nucleotides are, again, those little base pairs there. And then we have 10 million single nucleotide polymorphisms. And what the heck is a single nucleotide polymorphism? So I'm going to just jump to this. So single nucleotide polymorphisms are basically genetic variants that are quite common. So more than 1% of the population has single nucleotide polymorphisms, or I should say more than 1% of our genes have single nucleotide polymorphisms in them. So it's very, very common. It's older in evolution. They've happened over long periods of time, and it's just one nucleotide. So if we come back to this drawing here, where you look at the purple and the blue and the orange person. So the single nucleotide polymorphism there is just that one column where it says SNP. So all of the rest of those nucleotides are identical except for that one column. And you can see with this, if you have one column that's different, what's going to happen is it'll end up making the protein different. So if you look at down below, you could have a normal protein, say with the purple, and then with the blue person, with just one little difference, you could have an Alzheimer's variant. That's just one example of how a single nucleotide polymorphism can potentially impact what's happening with the protein formation. The single nucleotide polymorphisms, or I'm going to just call them SNPs from now on. So they're just weakly associated with chronic disease. So if you have a SNP, that does not mean you're going to get the disease. You usually have to have various multifactorial things happening at the same time in order to develop a disease from a SNP. On the opposite side, mutations are newer in evolution. We haven't been holding on to them as long. They are less frequent, and it can be big chunks of the nucleotides. And because it's a big chunk, that's going to have a really profound effect on the organism and disease. So mutations, we're not really talking about mutations when we're talking about how we're looking at genomics-based precision medicine. We're talking about SNPs. And SNPs, what we want to look at is, how many do you have and how are they going to come together to potentially impact your health and wellness, either in a positive or potentially harmful way. Now, when we look at the different SNPs here, we have so many genes that are related to disease. So 2,000 genes are related to disease. And here's a list here of all of the diseases, or many of them, most of them, that are related to genes. And we're learning more and more every day. But if you look at this, you can see that like almost all of the diseases that we can think of have some sort of genetic component. Now, is it some diseases are clearly genetic and others just have a mild impact. So what we're looking at here is there's everything, when we look at that big umbrella of health and wellness, genes and genomics are going to impact everything pretty much. So if we're not looking at these, we're kind of missing out on an opportunity to leverage the information to help optimize our health and wellness. Okay, so let's look at the single nucleotide polymorphism one more time, just to kind of get a picture one more time. So here's a visual of between the A and B there. If you look at A, the person has A and T with that little pair. And then if you look at B, they have G and C. And what's going to happen then is as that blueprint is being read, it's going to make a different protein. So imagine in a case of like, I'm making cartilage, for example, for all of my joints. And I actually do have some SNPs that impact my cartilage. So if I am making cartilage that's impacted and I have an altered protein, that's going to lead to me developing osteoarthritis as I get older if I'm doing other things that are going to impact my joints. And so does that mean that I'm definitely going to get osteoarthritis? No, it doesn't mean that definitely, but it does mean that I'm going to be more susceptible to that. So we can take this information and then support our bodies in a way that minimizes us having any sort of deleterious effects based on our genomics. Now, mutations, I just want to show this, mutations are much more significant. So when we get mutations, it can actually completely stop the coding of protein, which can be super harmful. Depending on the protein and enzymes, it can actually can be lethal. And so mutations are much more severe and they are much more obvious. What we're talking about are SNPs, which are more subtle and more additive with many of them. All right. The next thing we're going to talk about is how genetic variants and external factors are going to impact each other. So again, we're talking about SNPs and how they're going to kind of combine together and potentially make us more susceptible to disease over time. And an important thing to note is that genes have plasticity. And so what that means is it's never just the genes. So genes are just part of the story. So I showed this earlier, but just to revisit it again, if you have, it's a Venn diagram. So if you have one SNP that's weakly associated with the disease, another one that's weakly associated with the disease, and then environmental factors that are weakly associated, they all come together and then you get the disease. you can see if all you had was one of these three, or maybe even just two of these three, you're not going to probably get the disease. So it's multifactorial. And so the genes are only part of the story. The environment is only part of the story. It's how does this all come together? And these days we're just exposed to so much environmentally that our bodies just can't, we have not been able to evolve fast enough. So if you look at, the time that humans have been evolving as the length of a football field, 100 meters, what we're living in, with the changes with exposure to pesticides, EMF, all sorts of toxins, plastics, things that we just never were exposed to previously. That's like the last meter of the football field, maybe even less than a meter. And so our bodies just have not been able to evolve fast enough to deal with all these different stressors that didn't exist before. And that, I think, more than anything else, is why we're seeing like just disease. We're in a disease state in our country right now. And it's no one's fault. It's just we can't evolve fast enough. But what we can do is look at our genomics. We can look at what environmental factors we can change. We can look at which environmental factors we can't change, and then change what we can and support ourselves so that hopefully we're not getting this Venn diagram where they all come together. Let's talk about some specific examples of some SNPs with environmental factors that can lead to disease. So someone might have an insulin receptor substrate 1C allele and an FTO gene SNP, and they're eating a high carb, high saturated fat diet. So that significantly increases that person's risk of obesity and diabetes and all the things that come along with obesity and diabetes, including heart disease, strokes, et cetera. And so what can this person do? They can't change those SNPs down there. They're going to be there. However, they can alter their diet. So eat a lower carb diet, cut out the saturated fat, and then that's going to significantly decrease their risk by about a third. If you look at the diagram here. Here's another one. So I spent a long time in Juneau. I love Juneau. Juneau does not have a ton of sun, especially in the winter. And so someone lives in Juneau. They have a vitamin D binding protein, RS7041, and then they also have a vitamin D 25 hydroxylase CYP2R1. They're going to be at risk. So they've got two SNPs for vitamin D deficiency, and they live in a place where they're not going to get enough vitamin D through the sun. And so their risk of vitamin D deficiency is astronomically higher. Now, vitamin D, super easy. We can supplement with vitamin D. So this is something where we know that vitamin D deficiency is correlated with almost all disease processes. We've huge evidence over the past couple of years with COVID. We also know cancer, heart disease, autoimmune disease, pretty much across the board as we're studying it. Vitamin D is actually a hormone, and vitamin D deficiency seems to correlate with almost everything. However, pretty easy to mitigate this. We can just take vitamin D supplementation, and then it drastically decreases our risk. But if we don't have this information, if we don't know this, then we just are sort of going along and developing diseases and getting out of balance without even recognizing that we can pretty easily impact it. All right, now let's talk about the goals of genomics-based precision medicine. So the goal really, and how I look at it, is we want to identify our different SNPs that are relevant in how they impact or how they interact with the environment. And so I don't want to just know what your SNPs are. I want to know what your SNPs are and how you're living your life. So, and I don't want to just know how you're living your life, because it might be that, for example, that I can eat tons of saturated fat and have a keto diet, and I'm going to knock everything out of the park. And maybe you, if you eat saturated fat, you're going to end up with super high cholesterol and have a stroke. And so the environment, we need to look specifically, and we talk about this all the time. A lot of times we'll have multiple family members, and it's amazing. So some people, it's really deleterious, for example, to eat gluten, and for other people, it's not as big of a deal. So we want to look at, for each specific person, what are their SNPs, how are they living, and then how can we help them to live in accordance with their genes so that they're not going to develop any sort of diseases and they're going to thrive. And I think of genomics and precision medicine, genomics-based precision medicine, as I think of it as playing cards. So if 10 of us are playing poker and I have no idea how to play poker, but I get an amazing hand, and you know how to play poker really well, but you get a crappy hand, you might still win the game. And so it's not, what the cards we get, or, the blueprint we get with our genomics is only probably 10% of the story, if even. It's a really small part of the story. How we play the cards is much more important. And the environmental impact of on our genomics really begins in utero. And I think one of the biggest pieces of evidence for that is that identical twins, even though they have identical DNA, end up with different fingerprints. So this is kind of an interesting thing. Like, our fingerprints develop in utero in response to various factors, including hormones, stress that impacts hormones. And so you can have two twins, and maybe one of them is bigger, maybe one of them is getting a little squished, et cetera, and their fingerprints are going to be different. And that is environmental impact, changing the way our proteins are made in utero. So like before we're even born, and that continues throughout our body. Throughout our lives. So we can really impact how we're going to live, and how our health is going to be. We talk about health span versus lifespan. So if I live to be 100, that's my lifespan. I want my health span to be 100 as well. So I'm healthy up until when I die, when in reality, most of us are probably, our health span is maybe only 50% of the time. So maybe if we live to be 100, from 50 to 100, we're in disease span, and our bodies are not in balance and not working optimally. So really, it's as much, if not more, it's more about how you play your cards, how you live your life, than what cards you're dealt initially. And so, a lot of times people will be hesitant, I think, to get their genomics tested because they're worried that they're going to find out something and they won't be able to do anything about it. But 100% of genomic predispositions or SNPs are environmentally mitigated or environmentally impacted. And so I think of it as like, this is amazing information that then you can harness it to optimize your health and wellness. And now what I'd like to jump into is kind of looking at how we look at genomics at Boost, and how we use this amazing software from a company called Wild Health. And they were founded by a bunch of ER docs, some of whom I knew from training, and they've done an amazing thing of combining genomics with labs. So we look at what cards we were dealt and how we're doing today. Or another way I think about it is, you get your manual for your car. Say you have, like, I have a Subaru Outback. So say I have a Subaru Outback. I don't just bring the car to the shop and they don't just look at the manual and say, oh, based on the manual, we need to do this, this, and this. They're going to look at what's going on with the car now and then make changes based on that. And so the manual is kind of our genes, and how it's doing now is our labs. And so we want to combine that. So when we run the reports, this is kind of what the front looks like. And this is my report that I'm going to share. So I'm not sharing a patient's report here. So this is looking at all of my SNPs here for fat intolerance, carb intolerance, and saturated fat intolerance. And as you can see, I don't have the most amazing genes for all of these intolerances. They could be worse, but they're not amazing. Now, the oranges here mean that I got an allele from both of my parents. The green means I didn't get anyone, and the yellow means I got it from one of my parents. So I look at my genes, and I'm certainly at risk of developing things like metabolic syndrome, high cholesterol, diabetes, things like that based on my genes. But if you look down below at my labs there, which this was done last year, but it's pretty similar. I have been fortunate to live my life in a way that my numbers are not terrible, even though I have these genes. And so I have to be mindful. I know that if I changed the way that I was living, I probably would end up having quite high cholesterol and blood sugar and all of that. But the way that I have lived my life, luckily, has been congruent with my genes. We look at methylation. And again, I talked about methylation in the context of keeping our genes kind of tight and not reading genes that we don't want to be read. It's also super important for detoxification. Methylation is important for a lot of things. So if we talk about a lot in functional medicine, if we're not methylating well, it's going to impact everything. And so my methylation risks are relatively low, and I'm also, my diet has been such that it's been congruent with my methylation. So I'm doing okay here. We check labs like homocysteine, which are markers of inflammation and also methylation. And then TMAO there on the right, that's actually produced by bacteria in the gut. And so if I'm not methylating very well and I use methylation supplements, it can actually, depending on my genomics, depending on the bacteria in my gut, it can increase my TMAO, which is quite inflammatory. So we kind of look at big picture and more specific with this. These are looking at all my SNPs for vitamin and nutrient absorption. So I like to always focus on the oranges. So for me, in this case, you know, I've got collagen, iron overload, gluten, and zinc that I want to be aware of. So how has this changed my behavior since I learned my genomics probably a year and a half or 2 years ago? I know that I've got SNPs that make me predisposed to celiac disease and gluten intolerance. So I don't eat gluten anymore. I don't want to develop celiac disease. And I know that gluten is super inflammatory for me based on my genes and based on other tests. So I've just cut out gluten. I also know that I'm more likely to develop tendon problems based on my collagen SNP. So I make sure that I have bone broth or collagen supplementation every day. Iron overload. So I've only got one SNP there, but when I do ozone treatments, for example, I will dump some of the blood. So I'm minimizing the chance of iron overload. I think we think about not enough iron as being a problem and being anemic, but I think too much iron is probably actually more harmful. It's super inflammatory. So something that we don't think about as much in conventional medicine. And then zinc is so important for the immune system and for recovery. So I make sure that I supplement with zinc as well. So this is looking at my my genes for exercise. And you can see I'm kind of in the middle, maybe a teeny bit more strength, a teeny bit more slow recovery. But my take home for me is, for years and years and years, when I was running and competing in various sports competitively, I never took time off. Like, I thought that we were supposed to just exercise every day. And certainly that was what my coaches always told me. And guess what? I got injured all the time. So, you know, I think back to if I, you know, in university, when I was running, competing on a Division I team with cross country and track, I bet if I took a couple of days off a week, you know, and maybe did something restorative like yoga, but certainly not running or lifting weights, probably I would not have gotten injured and probably would have done way better than I did do. And so this is something that's interesting. I think it's hard to get this out of our heads. Like, I think we have this idea that more exercise is better. And there are some people that have fast recovery genes and that they're fine. They can exercise every day, but it does seem like the vast majority of people need to take more days off than they're taking, particularly in the places that I work, in the Tetons and in Moab. It tends to attract people that go out there and hit it hard every day, and taking time off to them will mean, oh, I just am going to go for a 2-hour mountain bike ride instead of a 6-hour mountain bike ride, when really what they should probably be doing is just going for a walk and doing some breathing exercises. This is looking at my sleep genomics. So a couple things that really hit me with that is I've got genes that make me more susceptible to things like developing cancer with circadian rhythm disruption. I'm sensitive to caffeine with sleep, and eating late also impacts my sleep. And so I have, you know, my sleep got disrupted from years and years and years of doing shift work. And I've used this information to adjust the way I live my life to optimize my sleep. And I do use an Oura ring to measure it's a biofeedback device to measure how my sleep is, my deep sleep, my REM sleep, and looking at heart rate variability. And I always recommend for everyone, for all of our patients, that we're collecting objective data. And so either the Whoop or the Oura ring really helps us to see, as we're changing variables, like, is it working or not? Because sometimes it's really hard to tell. Gluten is a common example that sometimes people notice a big difference and sometimes people don't. Gluten actually stays in the system for 7 months. And so you might not notice a huge difference, but maybe your heart rate variability is going up a little bit. Maybe your sleep scores are getting a little bit better. So I think any sort of objective data as we're tweaking variables is going to help us to get your body in balance faster. We look at neurobehavioral genomics as well. So looking at risks of things like anxiety or sensitivity to cannabis, which is helpful. Lots of people are struggling with anxiety now. And so we can look at, hey, maybe methylation, maybe if we increase your methylation, that's going to help with your anxiety. We look at kind of big picture here. These are all you can see lots of orange snips that I have here in terms of my genetic cardiovascular risk. So I'm just not someone that can get away with eating a keto diet all the time. If I did, I would probably get pretty sick. And that's just good to know. It's good information. This looks at, we look at the APO status. So ApoE3 is kind of the baseline genetic risk that most people have for Alzheimer's disease. ApoE4 increases the risk. And again, I think people think, oh, I don't want to know these things, but they are so modifiable. So if you have these genomics, we can help you to live your life in a way that you're going to really minimize your risk. We look at insulin resistance and SNPs that are related to that and how you can optimize your life to minimize insulin resistance. Inflammation, of course, is so important. We talk about inflammaging. So anything that causes inflammation is going to increase our aging and disease, basically. And so anything that we can do to decrease inflammation is going to be helpful. And I always like to bring up the example of vitamin E. So vitamin E for some people is really anti-inflammatory, and for other people, it's quite inflammatory. And by looking at your genomics, we can help you to determine where do you lie on that scale, and many other areas as well. The report gives a summary of all the different labs, and the red ones are out of range, the black ones are in range, and it allows us to follow them over time and hopefully get you into the black range for everything. All right. One thing I'd like to mention, we're getting near the end here, thanks for bearing with me, is your mind. We know that our minds are super powerful, and actually your mind can guide what your genes are doing. So I think that's super important to recognize. Now we know, of course, that people, for example, people who believe they're going to do better and have better outcomes with cancer are more likely to have better outcomes. But it's actually our minds are impacting what's happening on a cellular level. And so I think anything that we can do to help our minds be more relaxed and to help our nervous system is going to impact our genes, which is kind of mind-blowing to think about it. But we have lots of treatments that are very effective to help our minds to get into a more calm state that don't require 10,000 hours of meditation. And it kind of comes back to the relationship, the Venn diagram between the environmental factors and the SNPs. So if the environmental factor is that your sympathetic nervous system is super ramped up all the time, that can turn on genes. We know that stress can cause, and I shouldn't say turn on genes, but that can increase your risk of developing diseases when you have these predispositions. And so anything that we can do to kind of decrease our stress is going to make it harder. It's a harder environment for the disease to develop. And I think of stress as like a fire. And so if you're trying to put the fire out in your body to minimize the inflammation, to minimize the risk of disease, our minds and our nervous system are so important. And so we do stellate ganglion blocks, which is basically an injection in the neck. This picture of me doing one. It's an injection in the neck that's basically an autonomic nervous system reboot. We've got several webinars on that, so I'm not going to get into it in detail, but it basically helps to get your nervous system more balanced, more in parasympathetic, which is the rest and digest and healing side of the nervous system. Ketamine also amazing in terms of helping to balance our nervous system, which is going to impact our genes. So ketamine is going to increase our GABA, which is our chill out neurotransmitter. It's going to decrease our glutamate, which is our excitatory neurotransmitter. So the combination of stellates and ketamine are super helpful to get our nervous system in balance. And that actually does impact our genes, which is kind of wild. And the vagus nerve our stellate is our sympathetic nerve. Our vagus nerve is the longest nerve in our body, and it's our primary parasympathetic nerve. And if that's out of balance, that's going to cause everything to be out of balance. So the vagus nerve connects with the gut. the enteric nervous system, which is the nervous system around the gut, actually has more nerves than the central nervous system. So when the vagus nerve is not working and the signals aren't going as they should, that throws everything out of balance as well. And that also is going to impact the genes. And so it works in both directions. And that's kind of how epigenetics work. Like we have genes that are going to impact the proteins, and then the environmental factors are going to impact the genes. And so it works in both ways. We do retreats that combine both stellates and ketamine, and it's an opportunity to really change your life in a very short period of time. So you can come for a week and get some treatments supported with an amazing therapist, sound healing, craniosacral therapy, and integration, and the support of other people doing similar work. And it really just is going to, it changes what's happening in your body on a cellular level. So if you're interested in looking at your genomics, certainly you can call and schedule an appointment. We can talk about it. But if you want to just jump into it, the address to sign up is, you can look at it directly, or you can go to our website and you can click on the sign up for Wild Health Genomics. And once you sign up, they'll send you a kit where you can check your genomics, and then we'll talk to you about all the other details. If you've done 23andMe or Ancestry, you can also upload that raw data and save the $195 on the genomics testing. So after talking about all this, I'm curious if you could add, what do our genes impact into the chat box? And then maybe or Jesso, you can comment on anything that's coming in, or maybe anything that you're noticing. And then also, I would love to give an opportunity for questions, if there are any.
Great, thanks for your contributions into the chat, everyone. Seeing some beautiful responses here. Really just underscoring how much our genomics impact every area of our health. So it seems like folks are taking a lot out of this webinar. Also, a number of questions coming up. A number of questions around chronic injury and what the role is in genomics on managing chronic injury.
Yeah, great question. So it's multifactorial, really. So on a very simple level, if people have, I mentioned earlier, cartilage, if there's problems in sort of creating those proteins, that's going to cause problems. Also, if there's any SNPs that are going to make it such that the soft tissue, such as tendons and ligaments really, those connective tissues look almost identical under the microscope. So effectively, it's the same connective tissue. It's just, what is it connecting? And if we have SNPs that make it such that we're not creating optimal ligaments and tendons, then we end up having unstable joints. We end up having tendinosis. We end up having, ligaments like ACLs that blow out, and we just don't heal as well. So that's one level. And then the other level is if we have problems with inflammation based on our genomics and how we're living, that is also going to keep us from healing. So I think of, our body is like soil. And if the soil is like the dry desert Moab soil, it's hard for things to grow in there. And so if our body is like that and we get injured, and our body's like kind of trying to plant seeds to regrow the tissues that got injured, maybe it's not going to grow really well. And so sometimes our genomics are going to make it such that our soil is not optimal. Good question.
Great. Yeah, thanks for that. A question here about getting reports for kids.
Good question. So we can certainly run reports on kids. I think there's some ethical considerations around that. So we know also, I talked about the power of the mind. So, and that's something for adults as well. If finding out that, if a kid finds out that they have genes that make them susceptible, for example, to celiac disease, that can be really stressful. And maybe the family is able to support a gluten-free environment, for example, but maybe they're not, or maybe that feels hard at school, and maybe the stress actually is going to make it more likely that they develop the disease. People will ask, hey, I kind of want to run my genomics on my kid to see, like, are they better off being a strength or endurance athlete, fast or slow recovery? I think we have to be really mindful with this. It's not something that we should just do willy-nilly. I do think we're moving in a place where probably we will be checking more extensively genomics on everyone at some point in the not too distant future, and using that really as a roadmap for optimal health. But we need to be careful that we're doing it in a mindful way that's not going to cause more stress and potentially harm. So I think this is also new that we're figuring that out now. But the short answer is yes, it can be run, but I'm hesitant with kids, and like to have really mindful discussions with parents about it before doing that.
Great. Another question we have here is it possible to check genes without doing the labs?
Yeah, that's a great question. So for sure, there's lots of direct to consumer softwares out there where you can, 23andMe is one of them, and Ancestry, where you can check your genomics, and you don't check any labs, and that's that. It is possible, but I think you're really doing yourself a disservice. Kind of back to the idea of like the manual for your car. The manual for your car, like if your car is making a funny noise and you just read the manual, that's not going to necessarily give you the information. You want to do a diagnostic and see what's going on. And so I think the combination of the labs plus the genomics is really going to be the best foundation for helping your body to get in balance and in helping optimize health and performance. So, more and more, I really strongly discourage people from just checking their genomics, because it can also be really, it can be falsely reassuring. I've seen many people that have really great genomics in terms of their saturated fat, and carbohydrate intolerance SNPs, but they have diabetes or have really high cholesterol. And so if they're just checking their genes and not checking their sort of what's happening right now with the labs, with objective current data, I think they could be falsely reassured that what they're doing is just great. Good question.
We have another question here about genomics in support of managing chronic anxiety.
Oh yeah, great question. So I think that can come up in many different ways, but there are certain genes that specifically around methylation that COMT is one that is a common one that people have heard about, that if people have that, they can be quite anxious. That the COMT gene, I think of it on one end as the warrior, like WARRIOR, and on the other end, the worrier. And it's all about dopamine. And so on the warrior side, people, these are people that, can kind of go base jumping off of cliffs and kind of need to live on the edge to feel good. And then the other end are the worriers, who are more susceptible to anxiety, and they tend to be really high achieving people, but it can feel really uncomfortable. So methylation support can be super helpful for that. Other SNPs that can impact anxiety, certainly anything that's impacting the inflammation in general can lead to anxiety. And then really anything that's impacting the nervous system at all can impact, or the gut. So there's lots of different SNPs that we look at, and then we can kind of look at what support looks like to help with anxiety through a genomics-based lens. Good question.
This will be our last question because we're running out of time, but someone who's going through menopause, having questions about hormones and also the role of genomics in managing hormones.
Yeah, so great question. So hormones, I think I mentioned when I was talking about how the DNA is kind of the blueprint that ultimately leads to proteins. Proteins create hormones. And so if you have any SNPs that are going to impact the protein formation around hormones, that's going to impact your hormones. And then additionally, hormones are, the precursors for hormones are cholesterol. So anything that's impacting your cholesterol is going to impact your hormones. And so we look at all of that when we look at your genomics and your labs to see like where your genomics might be impacting your ability to have optimal hormones and how we can support that. And then just also reiterating that the environment is such an important role. And so, and that's why doing things like stellate ganglion blocks and ketamine drips seem to be beneficial across the board for almost everything. And it's because by getting that environment, the nervous system more relaxed, it's like getting the soil, like the Fertile Crescent instead of Moab soil, and then the body can get back into balance better or stay in balance. Good question.
Great. Well, thank you so much, Dr. Mulcahy, for this wonderful webinar. And we'll look forward to seeing everyone else next month.
Yeah, so thanks so much. And thanks for coming. And the next one is Tuesday, May 31st at 10 a.m. And I'd just love for people to write us or put in the chat box if there are any topics that you'd like to learn about. That'll help us to decide what we talk about next. Thanks so much.